Welcome to the Future Tech edition of the Finding Genius podcast. Forget frequently asked questions, forget common sense, common knowledge, or Googling for information. How about advice from a genius in their field instead? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond. They become very good at what they do, but only 0.1% are the geniuses of their profession. Richard has made it his life's mission to interview the geniuses of their fields in areas such as AI, 3D printing, quantum computing, blockchain and Bitcoin, and more. Don't miss out on amazing podcasts with geniuses. Review us on iTunes or wherever you listen, and go to futuretech.findinggeniuspodcast.com and subscribe today.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Lesley Cheng Sim. She's a research officer in the area of biochemistry. She has a PhD, has done postdoctoral research. She was awarded a Bachelor of Medical Science at Trobe University and honors from Monash University. A lot of great background in research. Now we're going to be talking about extracellular vesicles, which I've been talking about quite a bit lately, but the area is so big and so huge and so interesting, it's going to take countless researchers to figure it out. So, Lesley, thanks for coming. How are you doing?
Good, thank you. Thanks for having me, Fiction. I'm glad to see that extracellular vesicles has sparked your interests as well.
Yeah. I've spoken to actually probably over almost 24 scientists about it so far. But what got you interested in EVs, and what particular niche or part of the area are you working in?
Actually, I joined Professor Andy Hill's lab in 2011, and he started working on extracellular vesicles just a couple of years before that. But his lab was working on neurodegenerative diseases, and what was interesting that at the time, they started to find an enrichment of neurotoxic proteins in exosomes, which is one population of extracellular vesicles. And it sort of went from there, and the field of extracellular vesicles has really come to light in the last 10 to 12 years. And so it's a relatively new research area, and there's just been a huge exponential increase of publications in this area. And we've been lucky to be one of the labs who started it at the beginning. So it's been really fruitful. And when I joined the lab, I actually didn't know what extracellular vesicles was. During my PhD, I was studying neuronal death, and we did a lot of preconditioning treatments with the cell culture medium to see whether cell culture medium could rescue cells or stress cells in a neuronal death setting. And beknown to me that it could have been extracellular vesicles communicating and recovering these cells from cell death. So if I knew this during my PhD, it would have been wonderful. But now I particularly look at extracellular vesicles in a diagnostic way. That's my reason.
Well, okay, so what are you specifically trying to figure out about them? Is it EVs from a certain tissue? Is it what they contain? I mean, what are you trying to figure out?
Yeah, so in our lab, we have 3 major research programs. So we are trying to understand the role of exosomes in the pathology of neurodegenerative diseases, in particular Alzheimer's disease. And then we actually have several methods to be able to isolate exosomes from the blood. And we can use these as diagnostic tools to diagnose neurodegenerative disease. That's my main focus. And trying to exploit the exosomes as a vehicle to deliver therapeutics. So the 3 main neurodegenerative diseases we study in the lab is Alzheimer's, Parkinson's, and prion disease. And we have different clinical cohorts here in Australia and worldwide where we have collections, and we recruit patients to donate their blood. And we've been doing various discovery studies where we collect the exosomes from the blood, and we are able to profile the exosomes. So the exosomes mainly contain RNA and protein, and what we're particularly interested is the microRNA. So microRNA is a short 22 nucleotide RNA species that is that functions as a silencer in gene expression. However, they get packaged into exosomes within the cell, and then they get, as the exosome gets secreted from the cell, it ends up in the bloodstream. And by collecting blood and isolating the exosomes, we can basically crack the exosomes open and profile them using deep sequencing or next generation deep sequencing. So we've been
but who's sending and who's receiving in the diseases? What cell type's creating them? What cell type is receiving them? And then, if you're looking in the blood, there's tons of stuff in there from all kinds of cell types. How do you figure out what's yours and what's not?
So at the moment, we look at the total circulating population of exosomes. However, we can capture neuronal derived exosomes using antibodies specific to neuronal markers on the exosomes. So another aspect of our work is trying to also understand how these exosomes, for example, from neurons of the brain, bypass the blood-brain barrier to get into the bloodstream, where we can then isolate neuronal specific exosomes and have and determine whether they're more enriched in neuronal markers or neuronal specific microRNA that would give us a diagnostic read, more like a liquid biopsy of the brain.
Do you think you'd ever be able to culture a particular target cell type and, put them in some kind of matrix in vivo, see what exosomes they're taking in, take them out, crack them open and see what particular exosomes they've taken in? Is that a possibility?
That's a possibility. So in cell culture, we can dissect different populations of cells. So we can look at microglia, neuronal cells, astrocytes, etc. And then they will produce exosomes in culture, and then you collect those exosomes and do profiling on those. And then if you do, for example, RNA sequencing, you will see potentially many microRNA species that overlap across the cell types, and then you can find specific ones. But it's quite limited with this sort of technology, the depth of sequencing. So you might find specific or novel RNA markers across these cell lines, but it's just a matter of whether one cell line is expressing one particular microRNA species more abundantly compared to another. So there is some limitations to that. So it's quite challenging in that.
Well, how do you know that it's the neuronal cells that are giving off the exosomes you care to look at, for a particular condition? And again, do you know, like, you've identified for sure, okay, it's these cells as the creators, and then who is receiving them? Is that preferential? And how preferential?
Yeah, we could label them. So in a culture setting, you can imagine, even if we were culturing one particular cell type, one that in any one moment, cells are secreting exosomes, and those exosomes could be taken could just be taken up with the cell next door. So it's really hard to differentiate which exosomes being secreted, which one's being taken up. But potentially through labeling and microscopy, you could trace the exosome where it's going. Some labs have done microfluidic type of experiments where there's a channel in between two chambers, and they will seed particular cells, either the same cell type or two different cell types on one of the chambers, and then a different cell type on another chamber, which is separated by a channel. You will see a particular exosome coming through the channel and whether it gets taken up by the second chamber. So that's quite interesting, where you can study those type of...
Well, if we're to assume that exosomes are not just created randomly, they're deterministic, and the cell is creating them specifically inform another cell type of its condition or to instruct another cell type to do something, that tells you that there's, preferentiality in the creation and, the receiving of them. What about the mechanism by which, I know there's various mechanisms of the biogenesis of them. It's vesiculation and they bud off, or they come from the inside of the cell. But what about on the receiving end? Have you or anyone observed cells taking the exosomes in? And are there various ways in which they take them in? And is there a determination of what they will take in and what they will not take in? Or do they take everything in?
There's a selective mechanism. So there are, you can look by microscopy, for example, transmission electron microscopy, where you can definitely see EVs being taken up into cells. We do understand that there are several mechanisms that do allow exosomes to enter the cells, but actually we don't know how often this happens. So when we do treat cells with exosomes, and whether we label the exosomes and then put them on top of potential recipients, the uptake is actually surprisingly quite low. So we still don't understand the features or the characteristics that are required on an exosome that allows a cell to decide whether they should take that up or not. So that's still quite a challenging area and something that still needs to be further investigated. But for now,
does it seem like the recipient cell membrane has preferential sites that the exosomes bind to and then go through the lipid bilayer, or any guesses as to the mechanism?
In particular, in any sites, that's I'm not sure if there's any specific sites. In terms of microRNA, there is it's a really unknown area. So we're still trying to explore how these exosomes do get taken up by certain mechanisms. There are the classical ways. So you have endocytosis, you have the snare complex, the Catherine-coded complexes, very similar to the other type of microvesicle mechanisms that get taken up by the cells, or the snare complexes that you see with synaptic vesicles. So they're the same classical endocytosis pathways, but the most mysterious bit is really what is allowing which exosome to enter the cell, and what are the properties of the exosome that allows the cell to take what determines which exosome should be taken up or not. So that particular question is quite challenging.
Yeah, I mean, I've heard of like tunneling nanotubes that cells can literally connect to each other, and I heard they share organelles and all kinds of crazy stuff.
The classical way of cell-to-cell communication, we know prior to the discovery of exosomes, is the nanotunneling, where cells have this tunnel between them and they can pass proteins and RNA across to each other. Obviously, there's also cell-to-cell contact, and in particular infectious diseases, like the diseases we study, like prion disease, where neurotoxic proteins can pass from one to another via cell-to-cell contact. That's pretty obvious. But some of these diseases, certain organs from distant sites of the body get infected. So this is where we believe exosomes are. And my work really just looks at how we can use exosomes as diagnostic markers. So whether these exosomes are secreted from diseased tissue and then they enter the bloodstream, and the ability to be able to use them as a liquid biopsy and provide a diagnosis, hopefully an early diagnosis for patients.
So what is your particular area of expertise, you feel like, in the EV world? Is it being able to identify, given a blood sample, separate the noise from the signal and find out, okay, these microRNAs came from this cell type? What would you say is your focus?
Yeah, so one of the reasons why we use exosomes as our diagnostic tool is because we do find an enrichment of disease indicators in exosomes compared to, say, looking at neat blood. So one of my earlier studies was really to determine whether it was necessary to even go that extra step to isolate for exosomes, because practically in a diagnostic lab, the methods that are used to isolate exosomes, they don't have that sort of equipment in diagnostic labs. So to ensure that there was a reason that we should look into exosomes, I did a study on comparing the neat blood volume versus an enrichment of exosomes. So upon doing that and comparing the two different sample types, we could see that disease indicators, in particular for neurodegenerative, the disease indicators associated with these conditions were enriched in exosome samples. And by having them more enriched in exosome samples, the signal-to-noise ratio is much better. And so what happens when the signal-to-noise ratio is much better is that we get this concentration of these disease indicators. And when there's early pathology changes in the brain, these disease indicators are already present in the exosomes, whereas they would have been diluted in the whole blood if you hadn't enriched for exosomes. So by doing an enrichment with exosomes, we're able to pick up early indicators of changes in the brain. And this provides us an early readout of what could be happening in the brain and in these patients, and giving them an early diagnosis.
So what are some of the biomarkers that you're able to pick up, and what's the significance of certain ones being elevated?
Yeah, so what we did in an initial discovery study, so we had a cohort of patients that were clinically diagnosed with Alzheimer's disease, and then obviously a cohort of healthy controls. We isolated the exosomes from their serum and did next generation deep sequencing on their blood exosomes. And what we found was a panel of 16 different microRNA species that were either downregulated or upregulated in Alzheimer's disease patients compared to healthy controls. So those 16 biomarkers, we call them, they're associated with Alzheimer's disease. And so in various validation studies, we then got a new different cohort and screened them with these 16 microRNA biomarkers. And we've been able to obtain greater than 98% accuracy for predicting Alzheimer's disease in these patients. So now we are trying to move this into the clinic, but meanwhile we're also looking for panels for Parkinson's disease and also other neurodegenerative diseases as well, using the same technique.
Are you looking just for an increase in certain microRNAs? Is there a subsequent decrease in other ones that would normally show up in larger amounts in healthy people? Or is there just an increase in certain ones?
No, they're both. So some of them are an increase and some of them are a decrease. And what we find is that when we do so with the microRNA analysis, we can also do pathway analysis. So each microRNA can target several genes, whether to silence, in most cases, silence the gene expression or regulate gene expression. So what we found with the pathway analysis is that many of the microRNA that we picked up during the unbiased sequencing actually target genes involved in Alzheimer's disease, which was really great feedback in that those that were upregulated in these patients actually are associated with Alzheimer's disease. So back in the lab, to validate the function of these microRNAs, we are overexpressing these particular microRNAs in cell lines and having a look to see whether we can confirm that these microRNAs are actually associated with genes that are known to be involved in Alzheimer's disease, such as APP, that is involved in the creation of amyloid beta, which is the neurotoxic protein involved in Alzheimer's disease, and to see whether we can even package that particular microRNA in exosomes, redeliver it to correct gene expression.
How do you know that you're at the start of the chain or the start of the cascade? So these neuronal cells, they're microRNA expression profile or predominance profile is different. Some there's a lot more, some there's a lot less. But what caused them to have an altered, emission of exosomes? Can you go back one step further in the chain?
Yeah, that's an interesting question. We don't know what comes. From what we've seen is we can actually detect microRNA changes before messenger RNA changes, which then you would say that's where you start to see protein changes and then changes in pathology of the tissue, whether it's the brain or an organ, for example, in cancer. So what we've done we have several mice models, longitudinal mice studies, where we have done deep sequencing and looked at the expression changes across time. And what we see is what I just mentioned. We can pick up microRNA changes much earlier on before the changes in pathology are observed or even clinical symptoms in the mice. So that really helps us support the idea that our microRNA blood test is able to give us an early diagnosis of particular diseases. There are people who are looking at messenger RNA markers, obviously changes in protein, but actually what we're finding is we can look for microRNA, and that happens before everything else. And with the enrichment of microRNA and exosomes, that really gives us a preclinical diagnosis. So at the moment, we also have a longitudinal collection of these patients with Alzheimer's disease. So for the patients where our blood test has predicted them to have Alzheimer's disease, we actually have their blood going back 5-6 years ago. So these particular patients have been coming to the clinic every 18 months for the past 10 years. This is called the Australian Imaging Biomarkers Lifestyle Studying of Aging, ABLE. And so what we've been able to do is go back into the blood bank, get their bloods back 5 years ago, and determine at what point the microRNA expression changes do happen. And what we find is that we're able to actually pick this up 3 years before them presenting with symptoms. So that really gives us great confidence that we're heading the right direction with our blood test.
Do you think that there's so we have I guess I look at it as the cells that create the EVs and then the cell types that receive them and are altered by them. But is there is it circular? The target cells, are you looking at the EVs that they emit, that they give off, and do they have certain microRNAs or messenger RNAs that are now different, that affect the original cell that tried to influence them, the neuronal cell, for instance, and thereby changes them? Is it circular? Can you tell?
Yeah, circular. No, I don't think you can tell. The way I see it is if a cell secretes an exosome of microRNA, whether it's from a disease setting or a healthy setting, and a cell then receives it, it's up to that recipient cell to use what it wants to use from the exosome it's taken up. And either it secretes what it didn't need, keeps what it needs, secretes what it doesn't need, and passes it on to the next cell. That's a whole different other area. We haven't been able to see whether an exosome that gets taken up is then straight away secreted again. Because if you think about it, once it gets secreted, once it gets received by a cell, we don't know whether the whole exosome gets taken up or whether the lipid membrane just gets fused with the plasma membrane and the contents get emptied. And then once the contents are free, they're basically swimming in the cytosol. So how does that then get wrapped back up in an exosome and get secreted back out? So that's also a mysterious box that we don't know. It's hard to catch, even by microscopy, a full exosome coming into a cell and exiting straight away.
Yeah, you can't just use light microscopy and watch it, right?
No, so the microscopy is getting better every year. So we have super resolution microscopy, which is getting better as time goes on. And we're hoping to be able to utilize that technology to see whether we can see whole exosomes getting taken up. And that particular area is of interest to me because what we want to do is to be able to mimic a blood-brain barrier in vitro and image that, catch that actually happening. So we know that neuronal cells in the brain produces exosomes. We know we can capture them in the blood, but we've been unable to actually see the evidence that those exosomes are bypassing the blood-brain barrier. And it might be that in these diseases that the blood-brain barrier is just compromised and it's leaky and the exosomes go through tight junction gaps rather than going through a whole cell. So that's quite mysterious too. We really want to know what mechanism that allows an exosome from a neuron in the brain to fully come across the blood-brain barrier into the bloodstream. Whether that actually does occur or it needs the brain to be compromised for exosomes to actually bypass.
Yeah, gotcha. Well, lots of questions. Very good.
A lot of questions.
For now, we're just about out of time. What's the best way for people to follow your research and learn more?
They can contact me via email if you want to put that up on your website. You can just Google me. My scientific name is actually Lesley Cheng. So if you just Google me on that. I also have a TEDx talk that's on YouTube that I talk about my diagnostics there. So I am out there on the web. So happy for everybody to reach out in that way.
Okay, very good. Well, Lesley, thanks for coming. I appreciate it.
Sure, thank you.
You've been listening to the Future Tech edition of the Finding Genius podcast. This podcast is information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed. Review us on iTunes or wherever you listen, and subscribe today by going to futuretech.findinggeniuspodcast.com.